Thank you, Sophie, and thanks to Christine and Rare Cancers Australia for the chance to come back 10 years later to Can Forum, which has clearly become a very important vehicle for discourse that affects every single person in this room. I'd like to pay my respects to the Ngunnawal and Ngambri people on whose lands we're meeting today. I was honoured recently to have delivered the National Press Club address on Rare Cancers Awareness Day. In that address, I gave an overview of modern oncology, which today stands on the cusp of a once-in-a-century, generation-century change. That change is from a world in which cancers are classified by anatomy and the microscope to a molecular conception of cancer. It's hard to overstate what this means. The proof of science is in its ability to predict outcomes. That is the Enlightenment model. It's what's produced the world that some of us, are fortunate to be in today. If you think of the impact of a molecular concept of matter, Mendeleev's periodic table predicted the properties of matter that were absolutely unimaginable on the basis of looking at a rock or a piece of metal, laying the foundation for Niels Bohr's conception of the atom itself, and fundamentally changing almost everything about the way we see the world in the 20th and 21st centuries. So a molecular view of cancer has the same potential. As a genetic disease, a molecular view of cancer has directly and massively impacted drug development. The proof of that is simple. Clinical trials of novel therapies today, and this is data from actually 7 years ago, so it will have improved, are now 6 times more effective than they were in the 1990s when I was a young doctor. More than 90% of the world's cancer drugs in development today are rationally designed against a target by science. And the good news is that in 2018 alone, there were more than 840 molecules in drug development. These are the drugs of the future that make a difference to our patients today. And we can see the evidence of that in non-small cell lung cancer, where we went from no treatments, no targeted treatments in the 1990s, to 11 different classes of treatment, which cover more than 60% of the lung cancer population. And over the same period of time, we've seen an improvement of survival. Today, in what I think of as part 2 to that National Press Club address, I want to move from theory to practice. The time has come to act, but first we have to have a solution. And that is, I feel, as a geek, as the Scotty in the Starship Enterprise, not its captain, it's the responsibility to make the warp drive of this actually work. And I believe we've come to a time when it's possible. Now, Australia's challenges and opportunities have already been exhaustively documented. We don't need further information. Government papers alone include Alan Finkel's 2018 paper on precision medicine, Trent Zimmerman and Mike Freelander's fundamental paper, New Frontiers into Novel Therapies in 2021, the 2023 Treasury Intergenerational Report on Australia's Future to 2063, and this year alone, the Senate inquiry into equitable access to diagnosis and treatment for rare and less common cancers, and the Health Technology Assessment Review, MTP Connect's 2024 report on clinical trials, and Australia's Medical Science Co-Investment Plan. All of these lay out in a blueprint what we can all agree are our challenges and our opportunities. What I want to do is to reduce all of that to some fundamental principles that I believe must form the basis of a credible, a just, an effective and sustainable solution to implementing the miracles of modern medicines. The first principle is this: we must solve the emerging barriers and inequities faced by cancer patients in Australia today. We've heard about that all day. These include the length of time taken for access to newly effective therapies, the structural disadvantage of patients with rare cancers and other cancers as well, as I'll come to, the challenge of implementing access to genomic diagnostics as a routine, and the challenges of pan-tumor therapies. And there are many more than this, but let's start off with those as the challenges we face today. The second principle is that we must preserve the concept behind value for money. Taxpayer investments have many demands they must be transparently and fairly distributed. There is no future I can imagine in which this money can be allocated without reference to the many competing demands on those funds. The third principle is that we must address the long-term sustainability of health innovation. The intergenerational report has identified a future with an ageing population, a diminishing taxpayer base, the rising costs of healthcare as we age, and the need for workforce evolution to adapt to the new economies of the 21st century. The sheer pace of scientific progress is a complex system designed in the 20th century for incremental and empiric advances in medicine, not for the revolution now underway in oncology. And this is a big deal. Cancer is the leading cause of death in our communities. How can we do all of this while preserving the best of our current excellent healthcare system, which was designed to be stable, to be reliable, to do no harm, as Georgina said this morning? We propose a health system incubator, a structural entity that lies in the growing gap between the registration with the TGA of new, safe and effective therapies and the completion of value assessment that is adaptable to health more generally, scalable, and providing equitable access during all of this to all Australians, and that generates the data-driven evidence to support value and value for investment. I believe a structural solution is required to buffer the pace of science as it integrates health innovations into healthcare. It must provide desperate cancer patients with access to real advances in care. At the moment, that 450-day gap will see 11,000 rare cancer patients die in the gap between TGA listing and access to a treatment which will be released into the public as an abstract at, say, the ASCO in June of this year. They'll see the results on the screen and in the newspapers, but they'll have to wait 455 days. And that time is time that, as Rare Cancers Australia articulate, is time that cancer patients just don't have. So we need to solve this problem by, at the same time, offering good value for money out of fairness prior to the completion of health technology assessment. And that value assessment needs to continue to generate a universal metric by which equitable assessments about value for money can be made. And this model is not just for cancer. What we're talking about is going to apply to all areas of healthcare. It has to be adaptable. And so the health system incubator lies between the TGA and the PBS, providing immediate access to safe and effective therapies, generating real-world evidence that would substantiate investment of public money, using a pay-for-performance model in which the money that we as taxpayers provide is given to the patients that respond. Now, how much better does it get than that? You're only paying for somebody whose tumour shrinks in this model. This solution should solve the difficult challenges outlined above, providing immediate post-TGA access to new therapies while generating that evidence. And moreover, it should become a vehicle for increased global investment into Australian research and development, as well as healthcare, to grow our economy. Now, this is nice theory. I've talked about this before. And as in all science, these sorts of ideas need to be tested by tackling the challenges we face today. So how will we test this idea? Cancer, oncology, is the perfect test case for the reasons outlined above. The opportunities of current therapies, let alone the tsunami of emerging therapies, make this case urgent. Our data already show the survival benefits for Australian cancer patients. This is data generated by Omico. It shows the extension of survival for cancer patients coming into the programming, having genomic profiling, and then access to a matched therapy. You'll see an extension of survival for those who don't receive an unmatched therapy of 14 months. This is a population which has largely exhausted standard of care treatments. And you see the extension of almost a 50% extension of survival simply from taking part in these trials. And these are predominantly through trials. These are not proven therapies. They're just matched therapies. This is the principle behind the increased effectiveness of rational drug development. 37% of the patients that we've screened, and we've screened 15,000 patients to date, have had one of these biomarkers that would do that. But they can only access treatments for the most part through clinical trials because there is no standard of care. Think of the impact on rural and regional patients alone, having to travel hundreds of kilometres with metastatic cancer to take part in an ivory tower clinical trial, something we need to solve. And the area of oncology which has been most significantly affected is for rare cancer patients, as we've heard today, but also those whose site of origin is unknown, so-called cancers of unknown primary. They often get forgotten, the cancers of unknown primary. Imagine if you can't even put a label on something. The tumour's arisen, it's a lump, but you don't even know where it started. You can't even be given the standard treatments for a bowel cancer or a lung cancer because you don't even know where it began. And for both of those groups, genomic profiling, a molecular conception of cancer, could be completely transformative. But the question is, how do we turn that into something that will make a difference? What we have, at least, is the first principle. We have an unmet need and a tragedy. So now I want to talk to you about the treatments that are currently available today that could make a difference. This is not theoretical. I'm going to show you actual data. In addition to drugs in development, this is my sort of fundamental way of thinking about cancer drug development. That 30% to the top left is where we don't have targets, where research and fundamental discovery is required. Fortunately, that section has massively shrunk over the past decades. The second group, about a third again, are patients who are receiving treatments which are just being explored, where we don't have clinical evidence yet. The remaining 37% is where we generate the improved survival I showed you in the slide before. And they can be broken down into two groups. One of them is treatments that are accessible through trials, because we haven't yet completed the evaluation process of clinical trials. But there's 15% where the data is out, and the data is out not just for a particular cancer type, but across broadly all cancer types. And it's that group that shouldn't be in trials and should be available as a matter of routine care. That's 15% of cancer patients that could benefit from this. And this is a list of the 7 Food and Drug Administration approved pan-cancer therapies, which are, as of the latest, trastuzumab deruxtecan from AstraZeneca in 2024, April 2024. This is like 2 months old. This is a relatively new development. It only started in 2017. And all of these drugs, for the indications involved, have been shown to work across multiple cancer types. So these could be transformative for cancer of unknown primary and for rare cancers, because you don't need to know what the label is. You could reasonably assume that they will work regardless of what that label might be. So how good are these drugs? Or more precisely, how good by comparison to the gold standard of the drugs that we have currently approved on the PBS? So Georgina very kindly started off this day by describing this, and we saw the absolutely incredible progress in melanoma. I remember melanoma in 2001. We had dacarbazine with an 8% objective response rate, and look what has happened in the past 20 years for melanoma. Absolutely incredible through immunotherapies. So this is the data on which approval of pembrolizumab and nivolumab was made by the PBS to make it accessible. This is a gold standard of a drug that is effective. And this is what's called a waterfall plot. Above the line is bad, the tumour's grown. Below the line shows the degree to which the tumour has shrunk as a consequence of getting the drug. And you can see that both have this pattern where about a third of patients don't respond for reasons we still don't understand, which has been solved in part by adding additional agents. But 66%, 2/3 appear to respond. And the objective response rate here, the shrinkage of the tumour, is 43% in one case and 33% in the other. So this is our gold standard. We funded this, right? So we think this is good enough. These are the pan-tumour therapies. So apart from the figure to the top left, which is a bar chart, they're all waterfall plots. And the difference with the previous diagrams is that they've got different colours. And that's because there are different cancers. And you'll see that in every single case, there are multiple cancer types which show a fall below the plot, but below that horizontal line, as a consequence of being treated when you carry the relevant biomarker. And you have to get the biomarker, so you're replacing anatomy and the microscope with a molecular view driven. It's necessary that you have the genomic profiling, but you use those molecular profiles to identify patients no matter what the cancer type, and you can see the benefits here. All of them show effects which are at least as good as that for pembrolizumab and for nivolumab. The good news is this is just the beginning. So this is data that was generated in partnership through the help of Roche, where we looked at all phase 1, 2, and 3 trials that were going on in 2022 and projected on the basis of historic success rates the number of pan-tumor therapies out to the end of this decade. And in the blue, you see the ones that would be identified by a genomic biomarker, and it's projected to reach 30. So all of a sudden, you go from 15% of patients who would benefit from a pan-tumor therapy, potentially to filling in that pie chart with drugs that are going to work no matter what the label is. And remember, that's the secret. That's the secret that will enable us to treat patients. But the problem is that the individual targets for these drugs are rare. So you see in this column here the frequency of the target. And you see down the bottom NTRK fusions, 0.23%. I think that's an overestimate. I think it's probably closer to about 1 to 2%, which means you have to screen 1,000 patients to find the one patient who has an NTRK fusion. Now, the incredible thing is it's a 75% objective response rate. That's twice Pembro and Nivo. A brilliant drug, but it only works in the patients who have these NTRK fusions. And the problem is, if you're going to do a health technology assessment on this, it will cost you $2 million of screening to find the one patient that would benefit from the drug, no matter what the cost of the drug. And we believe the secret here is to combine these drugs, all 7 of them. And when you add up all these numbers, you get to that 15% figure. And all of a sudden, rather than $2 million to find a patient, it costs you $15,000. And by 2030, it'll cost us $3,000, where the drugs themselves cost, let's say, $50,000 a year. And just to emphasize those objective response rates, I still can't believe the most recent one, deruxtecan, 61.3% objective response rate against any cancer that has been tried with immunohistochemical staining. It's not just genomics, it's also protein-based tests, but for the most part, genomic screening. So this is the potential of the drugs to make a difference in the future. And this is what Prozia would test. What we're proposing in the Health System Incubator is to test these radical ideas of combining drugs together and sharing the cost of the screening to generate these health value assessments. We would take patients with rare cancers and cancers of unknown primary. We provide genomic profiling using the infrastructure that we've got. We allocate them to these 7 pan-tumor therapies, and we watch what happens. We collect the data, and we take that data back to our regulators and say, see here, this is value for money. That's the premise. If we can show that it's good value for money, why shouldn't we change the paradigm to make a difference forever for rare cancer patients and cancers of unknown primary, which kill 1 in 5 of our cancer deaths? And that's the ultimate game, not to bypass the health technology assessment system, but to adapt it for the 21st century, but to do so in an evidence-based way, and to create a system for doing it that allows you to replace this problem with any problem that is of such a complex scale, with such implications, that it needs to be tested at the system level rather than doing it through clinical trials alone. Now, this is a busy slide, but I'm going to give you a quick crash course in health economics. So do bear with me. This is very important because the punchline of this will establish the third principle, that this is good value for money. So, all of the above treatments are based around the idea that this will generate value for the PBS. Well, let's start with what's historically good value for money. Value is based around a single metric which enables evaluation of health benefit across any condition, and that metric is dollars per quality adjusted life year gained. It sounds complicated, but all it means is that the additional cost of what you're paying for is going to give you a certain amount of healthy time that will apply under any circumstances. It allows comparisons between vaccination strategies, health promotion campaigns, all the way through to high-cost drugs. In the past 2 to 3 decades, the magic number which we've used as good value for money has been $40,000 to $70,000 for each quality adjusted life year gain. Above $70,000 is considered debatable, although MSAC and PBAC do consider other factors such as unmet need or equity. Below $40,000 would be a no-brainer. So we've undertaken modelling to test whether everything what I've talked to you about today would be credibly true. We've done it across the 7 matched therapies and included the cost of genomic profiling. And the punchline is that our base case scenario, independently reviewed by a health economist, is that it would be $45,000 per quality gained, with a best case scenario of $37,000 per quality gained, and a worst case scenario of $73,000 per quality gained. Most importantly, the total likely budgetary impact under the MBS and PBS for provision of CGP to everybody with rare and cancers of unknown primary, and access to the 7 life-transforming therapies I've just shown you, would be less than $80 million per year. To put this into context, This is 3% of the current $2.3 billion spent on the top 25 high-cost cancer drugs in 2020, for a population that accounts for 20% of cancer deaths. And it includes the cost of screening. Now, there's no doubt uncertainties that need to be resolved, and that's the purpose of PROSI. We need to be sure about these estimates, but at least the question is plausible. So to finish, we have an unmet need, we have systematic disadvantage, we have effective therapies, and it looks like we have reason to believe that this will also be achievable with traditionally acceptable standards for value for money. This is a Rubicon moment, starting with rare and CUP patients, solving forever decades of disenfranchisement and disadvantage. From this point on, any patient with a rare cancer will be at the front of the queue rather than the back of the queue because they'll have genomic profiling on tap. It means they'll be the first population that you progress drugs in. It will incentivize companies to pursue small indications with massive benefits because cumulatively, over the next 20 or 30 years, it's the sum total of all those individual interventions that will actually get us to that 90% cure rate, the 90% 5-year survival. And where do Omicron and Zero fit in? Over the past 6 years, our organisations have invested more than $300 million in developing a trusted national infrastructure with a proven track record that can ensure equity of access. We are honest brokers that can assess tumour response, thereby enabling pay for performance during PROSI. We have established a model that allows global industry to invest in Australian clinical trials, thereby growing the economy, injecting resources into the system, creating jobs. And that means an additional 20% of rare cancer patients will access trials. We've still got that 20% of the pie that needs to access through trials. It's not just the 15% that we put onto the PBS, it's the other 20% that we also need to ensure get access to these emerging promising therapies. The health system incubator can support a forward-looking but equitable healthcare system in which research becomes a standard of care, where all stakeholders contribute to ensure faster progress, reduce the costs of drug development, and address long-term sustainability. Thank you.